Okay, so we will be going over chapter 18 today. This is talking about the different diagnostic methods. And again, I want to say a lot of this, a decent portion of this is going to be stuff that we've already learned about, and we're just applying it to the diagnostic setting. So it shouldn't be a whole ton of new information. What I will say is that there's 3 categories that we have to put everything in. Well, there's 4 technically, okay, because there's an other as well, but that's like imaging and things like that. So the random ones, but our diagnostic tests that we're going to be learning about are either going to be phenotypic, immunologic, or genotypic. So they have to fall within one of those 3 categories. And so if I ask a question on a test, which category does biochemical testing of your bacteria, like we're doing in lab, which is a diagnostic test for diseases, which category would it fall into? The stuff that we're looking at is all like pH dependent based on if you can ferment or if you can use this molecule to do that or whatever it is. None of that is immunological. That's not antibody or antigen response. And none of that is genotypic. We're not looking at the exact gene sequences of these guys as comparisons. So that's all going to be phenotypic. So basically what that organism is capable of doing physiologically falls in that phenotypic category because that's the enzymes they're expressing. That's the things they're able to do. So we're going to talk about those. But that's sometimes an issue that people have a hard time with. Maybe categorizing biochemical tests as phenotypic might not seem intuitive at this point, but if you know that there's 3 main categories and you know that it's not immunologic and it's not genotypic, that should narrow it down. Point of care is another thing that this isn't a category, but we need to consider the fact that some of these tests are now performed at the bedside by nurses, or even in veterinary cases where you'll have like these cartridges you can put into these like handheld devices that'll read out blood samples right there instantly, pretty much, or it might take a couple minutes, but it's pretty close. Or little devices they can have like at clinics and stuff that you can put it right there. What I mean is you don't have to send it to a lab and like wait for the lab to run the test for you. If you're working in an ER, ERs will order tests, or most of their tests will be coming into labs as stat. That means they have priority. It does not mean that the second that the lab gets that test, that it gets put on the machine. It absolutely does not mean that. What it means is that yours will get priority over the floors, and that's it. If somebody else sent a stat sample before you, their stat sample has priority over yours. So we can't just constantly be putting the first, the next stat sample that comes out from the ER onto the machine before everybody else's. So that is only going to be prioritization. Whereas if you want to have an idea of if your patient is experiencing symptoms from kidney failure versus maybe some sort of liver issue versus some other sort of toxic aspect of their blood, you can run one of those point of care diagnostics with like a finger prick, as opposed to like drawing whole vials of blood, sending it to the lab and waiting the hour that it's going to take for those results to come back. And so they might not be as reliable as the blood tubes sent to the lab, and they might not give you as many of the specific details, but it can at least paint you a picture that this person is having kidney failure as opposed to some other physiological thing going wrong. Because all you'll see are those visually presented symptoms, maybe low blood pressure and things like that. That'll at least give you an idea of that. So you can do point of care stuff at bedside, running BMPs and CMPs and CBCs, or at least hematocrit, something like that, to get an idea of that. So if your patient is looking pale, if your patient has just been really tired and everything, and you can run at their bedside a hematocrit and see maybe they're anemic, or what if it's something else that's going on with their body. So it gives you that sort of stuff. So that's what the idea is with point of care. I don't know how all of the other hospitals did it, but at Southwest, they kept all of those like cartridges in the lab, and the nurses would call up and order like a box full. And so we'd only have to send it to them like once a month. So for the most part, they would have those available to them for use at the bedside of the patient, to get those kinds of pictures, to let them know, do we need to order more blood work on this or not? All right, obviously, if you have somebody coming in with more serious issues, you'd want to get the blood work, but is, not everybody that comes to the ER is having a serious issue. Actually, probably 90% of them are not. All right, so this is our little chart of essentially what our tests are going to be like targeting. So here we're saying phenotypic being things that are observable. This is going to be our color changes in our physiological responses, our biochemical tests, and things like we're doing in our lab with our bacteria. We also have immunological. That's going to be always associating with antibody or antigen response. That includes T cell responses as well, which I know is not antibody, but it is immunological, dealing with antigens, right? And then we have genotypic. This is like looking at the actual genetic sequences of things, not looking at their expression, but the actual genes in the background. So A, T, C, G of things. When you're collecting your samples, you might want to think about how to collect your sample or what sample you need for doing a test. Obviously, if you're trying to determine if your patient is having some sort of, kidney failure, you might want to get a blood sample and look at, levels of certain things in their blood, as opposed to collecting like a fecal sample, which, yeah, obviously that makes no sense. So that's all that this is talking about. Most of us know what most of these samples could be, whether it's urine, feces, or how we might collect the blood or the urine, whether it is clean catch or if it is collected via catheter, sputum versus saliva, and cheek swabs, things like that, biopsies, all of that stuff. This little swab down here that I have the arrows at, this is pretty common thing that you're going to see if you are collecting microbiological samples, where you're going to have that red twist cap that'll break a seal whenever you go to collect your patient sample. If you're working in any hospital ever, if you are a nurse and you're going to collect a sample by a swab and that seal is broken, you do not need to be using that swab. That seems common sense, but people will like go over to like get ready for a patient and they'll have cracked the seal and not have swabbed that patient, and then think that they should be able to go use it for the next patient because they know they didn't use it. But that's bad practice, because if you get in the habit of that, somewhere in the middle, you may have used it and forgotten and not labeled it or something like that. So no, if you're going up to a patient, you get a sample, of course, have a brand new one. There's a swab inside of these guys that you'll pull out, swab your thing and put it back in. Sometimes there's like goo or liquid at the bottom that'll keep that bacteria happy until it gets to the lab. Sometimes there's a little like vial that you'll have to crack that'll release that liquid to make it happy. But whatever it is, there's usually some sort of transport medium in the bottom of that to keep the bacteria alive until it gets to the lab, because a lot of, not all, but a lot of hospital labs, when they get cultures, they aren't going to be like getting it up out of their little delivery system and going and swapping it immediately. It's going to sit on a shelf for a little while. And then when they get to it, they will go and swap the plates. And that's just because you only have a limited number of people that are able to do these things at once. So that's how that'll operate. And then when we're looking at your it's in there.
Oh, and it's in the grate?
Yeah, it's like behind this wall. So either that door is locked and I don't have a key to it. So that's how it's going to, yeah. I don't blame you for being an iPad. But yeah, that's how it goes. Unfortunately, today it's cricket day. Yes. So also, I mean, I know most people aren't here, and I'll probably say it again when we do the presentations because everybody will be here. But since I'm thinking about it, since the cricket annoys me too. But the other thing that annoys me about this room is the internet. And I'm sure you guys noticed that. We will probably just go ahead and have our exam in the other room on Unit 3 exam, just so that we don't have issues with internet with you guys. That way you can know that that's going to happen, and we don't have to just move and have everybody be like, great, I sat down and got everything set up and now I have to move to another room. So we'll just anticipate being in the room over there that we were in last time, so that we don't have to deal with internet issues. Because this room, I don't know what it is. It's some sort of fortress against internet somehow. It's the most unreliable room for internet access. Whatever, we just won't mess with it next time. All right, anyway, so moving on. When you're ordering your stuff, 99% of the time as a nurse or as vet, you're probably going to have the order made by the doctor or the actual vet doctor. And you're going to be just going through with collecting the samples and fulfilling that. And then the doctor, of course, might be the one interpreting the results. But as somebody who's the person that is most face-to-face with the patient, whoever they might be, you're the one who is going to want to probably understand what you're doing with that sample, why you're getting that sample, why are we looking for this sample for this patient, what's going on with my patient. You're not just there to be chatty and happy and collect the blood samples, right? Because of course, if you are there dealing with that patient or that pet, you're the one that's going to be with them the most. So you would want to be able to answer their questions as well as interact with the family appropriately. So the idea is that if a doctor orders a CBC, you might want to be aware of number one, when I keep saying to you guys CBC or CMP or BMP, if you don't yet know what those things are, even in a vet clinic, you're going to have those terms presented to you. CBC is talking about a complete blood count. That's going to be something that's looking at your red blood cells, your platelets, and all of your white blood cells as well. All the counts of those, as well as your hematocrit, your hemoglobin, all of that stuff. So giving the blood picture of things. Whereas CMP and BMP, MP in those are metabolic panels. So BMP is the basic one, CMP is the complete one. It just looks at more things. They're looking at liver enzymes and, products that are made associated with like kidney function and things like that. And there's all sorts of different ones that are going to relate to that. It'll also be related to like glucose levels in the blood and that. So it gives a picture to the doctor of their metabolic standing. So if you have somebody that you think is having some sort of kidney failure, you would want to get a CMP in order to look at all of those factors, whatever. But when you get those orders in from your doctor, whether it is an Epic or it is on paper, I don't care, whatever it is, it's going to say CMP. And it might, if you're lucky and you're working at a place that uses Epic or someplace like that where it's up to date, if you're using paper, it's probably just going to have that doctor's scripturally little handwriting that says CMP on there. And you're going to have to know what color tube to draw, how many tubes, if it is multiple tubes, because some of these are panels that you might have to have multiple tubes for and whatever. That's all on the phlebotomy side of things. If you're working at a hospital like Mercy, you might have floor phlebotomists that will be drawing these things for you. Like I said, if you're lucky. But if you're at Integris, you as a nurse will be collecting these samples, typically yourself, or you might have like paramedics and stuff that work, hospital paramedics and stuff like that, that'll work as phlebotomists or like assigned nurse phlebotomists that will be going around like they often have at the ER at Integris Baptist. They have like the nurse that goes around and starts the IVs and gets the, phlebotomy samples and everything, but you don't see that nurse. And it's a nurse, but you don't see that nurse ever again. They just come and get your samples collected. And then you have like your room nurse and whoever that comes in and deals with everything after that point and all that. So they have their systems. Everybody has their systems. But if it's in Epic, what happens is the doctor will put in the order for that BMP or whatever, that CMP, and then you as a nurse will see that in your patient's orders. You get to print that little label out and get your tubes together and come to your patient's bedside and collect those things through their IV or by venipuncture. And then be labeling your tubes bedside. And it should be happening. You collect your tubes right there in front of your patient and labeling it, showing it to them, saying, this is your name, right? This is everything on here is you. The patient verifies that. And you'll be verifying your labels beforehand with them, everything. So there's like cross-check after cross-check after cross-check, making sure that sample is being done correctly. Most hospitals are going to have you draw your sample with unlabeled tubes. Yes, that is correct. Unlabeled tubes going into the actual venipuncture line or the Vacutainer needle. And then you label it in front of the patient because you do not want to pre-label your tubes because you don't want to mix it up with other patients that you're dealing with because, hey, you're not dealing with one patient. You're dealing with a whole floor of patients, right? So, yeah. So you don't want to mix those up. So you want to label it bedside while you are drawing that. So this form goes along with all of that because all it's going to have on there is like a checkbox that says CMP. If it's paper, that'll just be checked. If you're on Epic, it'll just print out the thing saying CMP. Again, it is not going to explain to you what CMP means. It's not going to explain to you what tests are involved in a CMP. A CMP is not one test. It is many tests. And so being aware of the forms and how they operate is stuff that you'll get training on when you go work in hospitals, if you haven't already been working in hospitals. If you are not working directly with patients or seeing or interacting with their results, it still might seem foreign to you, even if you are working in hospitals or nursing homes or whatever. because this stuff is it's just one of that, it's the type of thing where you have to be trained on the procedural side of it so that there's not mix up between patient samples. But labeling, knowing what your tests are, all of that stuff is just as important as interacting with the patient. Because if you mix up patient samples, that can become your job and that person dying. So it's important stuff. All right. Yes, and obviously as nurses or working as vet techs are going to be more hands-on with their, patients, their furry patients, as far as what their results are going to mean and interacting with the tests themselves. You might actually be looking in the microscopes and diagnosing certain things like stool samples or whatever. You'll be doing those things. Nurses at hospitals, especially the bigger ones anyways, working in small rural areas, you might interact more with the testing. But if you are working at Integris or Mercy or someplace like that, Norman Regional, then you, or OU, wherever it is, you are going to be relying on the lab to do all of that work. And that's kind of why you have to come in and do this stuff with micro, is so that you can know what the lab is doing with your samples. It's great to know that you're going to take a swab and send it off and you never have to think about it again. But not understanding why they're doing that test or what that test means to your patient, that's poor medical practice. So you want to be aware of everything that's going on with your patient as much as you can. You might not be the doctor, you might not be prescribing the tests, you might not be the one making decisions afterwards, but in order to communicate effectively with your patient and tell them what's going on and why you're doing the test, and with the family members who are usually more irate than the patient, that you should be educated on those things. So anyway, sometimes when we collect our microbial specimens, typically you're going to have an idea, not always, of course, but typically you're going to have an idea of what you think is going on with that patient. They come in with, an infection in their skin. They've got a boil going on that hasn't been clearing up and it's been there for a long time. You might suspect MRSA, right? That's pretty typical suspicion. So if you think it's going to be some sort of staph infection, you might want to reach for selective media that selects for staph, because you already think that's what it's going to be. Might as well start, if we're going to take a skin sample or swab of a wound, you're not just going to pick up that infectious bacteria, you're going to pick up other skin bacteria. So let's go ahead and select for the thing we think it's going to be anyways. That's where your selective media that we've been learning about comes into play. We actually use that in the lab. Your doctor will order that it be cultured on mannitol salts agar, very commonly used for skin infections in labs. So they will order culture on mannitol salts agar, then the doctor will get back the results that, it produced acid. So that meant that it was fermenting mannitol and that it grew, of course, on that agar, and whatever that tells that doctor. But this is probably Staph aureus, and that we probably need to be treating it as such. So selective media is for situations where you think that it's probably this group of organisms. So let's try to, grow it specifically, as opposed to growing everything and trying to have to isolate out and do all the stuff that we have to do with complete unknowns. Differential media, again, tells you information about your organism as well. Maybe you think it could be this kind of gram-negative organism, but you're not sure if it is organism A or organism B. But organism B ferments lactose. Maybe you'll grow it on McConkey agar and see if it turns pink or not, So you could do that side of things as well. So selective and differential media might be used right up front for that sort of stuff. Whatever you are doing to try to approach to determine what your organism is for your diagnostic process, and even when it's not bacterial, but of course we're focusing on bacterial stuff here, you can use the theory behind the dichotomous key or an actual physical dichotomous key to determine what your organism is. And I've shown you guys the picture of the dichotomous key, I think, of what we've used in past semesters. I have to make a new one for you guys because you guys were lucky enough that I got organisms to grow better, because the pH of the water used to be 3 and now it's back to what water should be, 7. So now things grow the way they're supposed to, but I don't have charts that will accommodate the organisms you have. So you guys don't get your charts until the end of the semester. But basically what it will be is one chart for your gram positives, one chart for your gram negatives. And then after that, did your organism ferment glucose? If it did ferment glucose, which you would have acid produced, when it fermented it, did it also make gas? If it did those things, then did it also hydrolyze starch or urea or whatever it is? So these charts are set up in a way that you can say, okay, when I did this test, I got a positive result. So now I go down this route of the chart. So here we have starting off with our cocci at the top. Oh, up here, if you looked in your simple stain and saw that there were cocci in the image, well, was it gram positive or gram negative when you did the gram stain? If it was gram positive following that route, was it positive for catalase or negative for catalase? Or did it have these other like arrangements associated with it? And then based on the arrangements and whether it was positive or negative for catalase, which we learned about catalase, right? That was hydrogen peroxide being converted to water and oxygen. It's a test we're going to do at the end of our lab stuff. But if you get bubbles when you put hydrogen peroxide on your bacteria, you are catalase positive. You broke it down into oxygen. So that's all you do with it. Take your bacterial sample, look at it with a gram stain, see if it bubbles with hydrogen peroxide. Then you might see if it is a strict aerobe or a facultative anaerobe. If it is able to ferment something, so make acid in one of our phenol red carbohydrate fermentation tubes, for example, like those 4 that were all the same but different carbohydrates in the lab. If it's able to do fermentation in any of those, then it can ferment. If it can ferment, it is a facultative anaerobe. So that tells you something about that organism there. So if you followed it all the way down from cocci, it was gram positive, it's catalase positive, but it fermented, so it's a facultative anaerobe, then that would lead you to believe that it is Staphylococcus planococcus as opposed to Micrococcus or a Streptococcus organism. So Staphylococcus being the genus, Planococcus being a genus. It's not an organism by itself. But yes, so that could help you find that grouping, and you can do more tests to find out the exact species beyond that. But that's how a dichotomous key will work. That's how your dichotomous key is going to work. And it won't have like, I feel like ours are way easier to use than these personally, but it has like 1 test on it. It doesn't have like 5 on one thing, but so whatever. So that's the idea with the test we're doing in lab. 2, it's like whether or not, God, that stopped. Whether or not it's going to be positive or negative for tests will send you down different routes on your dichotomous key because it tells you information about that organism so you can like categorize them and finally weed it down to the specific organisms. So all of our biochemical testing that we are doing is based on physiological reactions by your microbes in response to the media that you're growing it in or on. And these are all phenotypic tests. So these are phenotypic, because we're looking at the actual expression of the genes and what results from that. So these are often visualized by color change. You put your microbe onto your media, and whether or not it produces an enzyme determines the color change that you see, or if you don't see one, or if it grows or not. You can do these tests en masse on these cards, these API cards that we can order, where like all of the tests that we're doing in labs where we have, oh man. So if you look at our test that we inoculated last time, that would be our fermentation tests of those 4 different carbohydrates. We had the MR test and the VP. Those are all in 1 tube, but we're going to separate into 2 tests. That's 6 tests just right there, MR, VP. And then the litmus milk stuff. That's a lot of stuff going on with that as well. But we could separate those out on individual tests. We have all these other media we're going to look at, like citrate, phenylalanine deaminase. We're going to look at sulfur reduction eventually, and whether or not you can utilize tryptophan and all this stuff. You can put each of those individual tests in one of these little like wells, these pill-shaped wells here. These are just wells where people put their bacterial culture into these little wells, like not even a milliliter in there, and incubate it in their little incubator overnight. And then they bring it out and look at their color changes on here. They can even do anaerobic tests by putting oil in the little top of those wells to block out any air from getting in there. So these API tests do all of the stuff that we're doing in our second half of the semester, all on 1 card in 1 day, basically. They are extremely effective for microbiological laboratories and hospitals. And this is what they're going to use as opposed to the direct media that we're working with in our lab. They're not going to be culturing the tubes. They're not going to be streaking slants or plates a lot of the times. Sometimes they will if that's just what is easier. If they just need to do 1 test, yeah, they're going to do the 1 tube. They're not going to do a whole freaking card because these cards are very expensive. And that's why you guys don't get to do them. just being completely honest here. So these are very expensive to run on these and order these. And you have to have like a subscription to their system because how, of course, they analyze it is, you can't just get the information by doing that and look at it and say, yes, we are positive. They have to get their results and their colors and what their colors say are positive or negative. And then you make these little like number systems for it and add it together and type in a number into their system. And it tells you this is E. coli. So that's how those things work. But it's very useful for microbiology labs at hospitals where you are not sure what your organism is. If your patient comes in and they're sick and you have no idea what it is, you can't run PCR on that. Why? Because you don't know what PCR test to run. What primers do you use for that if you have no idea what your patient has? So you start with something like this here, especially if you already know it's probably bacterial, and you can verify that with a gram stain. Then you can run one of these tests and get a profile, and it's going to tell you it's probably E. coli. Now you can go do your PCR test and confirm that it is E. coli for that patient, so we can start treating them appropriately. So this is really useful for stuff like that. This is what I'm going to be ordering for the cadaver testing, just FYI, because I don't have time to go through every single little test that we want to run. And we just don't have all of the tests. Like when you look at this card, y'all, that's 50 tests. It's called an API 50 because it's got 50 tests. That's just for lactobacillus species. So if you're this, you have to know that you're looking for lactobacillus first. That's a problem if you, so you can imagine how many tests and how many cards are out there. So already we have to have some idea of what route we're going down. And you guys are just doing like very basic stuff in our labs, looking at that stuff. And so if we're really looking for true unknowns where you don't even know where to start, you might be using several of those cards, unfortunately. So anyways, another thing you can do to identify, other than our biochemical tests and our en masse biochemical tests, will be antimicrobial susceptibility, not just to determine which antibiotic to use, which we already talked about, right? We know that's what Kirby-Bauer is really for initially, but also some bacteria have certain profiles that you can expect how they're going to react to ampicillin and, clindamycin and whatever SXT, whatever else you can think of. And put those discs on there. And then based on how big their zones of inhibition are, or whether they even have a zone of inhibition, will tell you potentially what that organism is. This is not used very commonly for general identification of organisms, but telling the difference between species and subspecies, it can be useful for some genera. So they say they use this for Streptococcus, Clostridium, and Pseudomonas species to tell the differences between these guys. We don't use it a whole ton. And we also don't use a ton of phage typing for identification. And this is really only useful for certain kinds of bacteria as well. So they use this for salmonella. If you guys get salmonella poisoning, there are so many different kinds of salmonella out there. It is nuts. So most of the time, if you're sick with salmonella, you have Salmonella enteritidis. That is a very general species name. But if you have Salmonella typhi, Salmonella typhimurium, Salmonella, there's another one, enterica. There's all these different ones, but those aren't actually species of Salmonella. They're like subtypes. They're subspecies of Salmonella enteritidis. So if you want to know what kind of Salmonella you have, sometimes when you have Salmonella typhi versus typhimurium subspecies, they might be susceptible to a phage type that the other subspecies isn't. So a phage, remember, these are viruses that can infect certain kinds of bacteria. And they only infect usually one species or even a subspecies. That's just depending all on those surface proteins of how they can get into those bacteria. So they only recognize those. So you put your patient sample out on a plate and divide it into little parts. And then you put little tiny amounts of little dots of bacteriophage in your little quadrants or whatever, right? And in that, you'll come back the next day and see where your bacteria grew on your lawn, and wherever there is a clearing area, just like we had with our Kirby-Bauer discs. Instead, we're using, instead of antibiotics killing your bacteria, we're using bacteriophages to kill your bacteria. So you'll have a clearing area where you don't have living bacteria anymore because the viruses will burst those bacteria open. So that's phage typing. We don't use this a lot, mostly for salmonella identification and comparing salmonella to like E. coli, because a phage that infects salmonella won't infect E. coli. And if your patient has diarrhea, yeah, it's just easier sometimes to do it that way if you have that available to you. So it just depends on what illness you think a patient has, what organism is causing it, which test you're going to be reaching for, and what's easier in that lab and in your setting specifically. If we're doing cultures of a patient's stool, let's say you have diarrhea, okay, and you order a culture of that patient's stool to see what it is. Well, if we're just straight up culturing that and looking at it, and we find out that there's E. coli in there, there's like some E. coli that's in that stool sample, but might not be the majority. There's some colonies that we got grown up in your stool sample that were E. coli. Does that mean that you are infected and have a disease caused by E. coli? Probably not, because everybody has E. coli in their colon. So how do you know that that is the E. coli causing that infection? There's just a little bit. It's probably normal biota. However, if you culture somebody's stool and it's like overgrown with E. coli, that's a problem. That's probably an infection. There we have overgrowth of something that shouldn't be overgrown. Same thing as if you look with C. diff, you might have certain colonies of C. diff, but if it is like completely just all C. diff in your culture, that's probably your culprit for that stool sample. Having said that, that's saying if you have just a few colonies of E. coli, that would be normal biota. If you have just a few cells in your sputum sample that are staining acid fast, that is not normal. Nobody should ever have any acid fast bacteria in their sputum. And we've talked about that, right? Mycobacterium species are not part of your normal biota. You shouldn't be coughing up tuberculosis bacteria if you're a healthy person. So if you've got tuberculosis bacteria at all, even one cell in your sputum that stains acid fast positive, then that is an infection and that is a concern. So comparing it to what you expect from the normal biota can also give you information about what's going on with your patient. Shouldn't even have any of that mycobacterium. Problem with these phenotypic methods is that they will rely on growing up cultures sometimes, which can take 24 to 48 hours. I say 24, it says 18 to 24. If you have E. coli and you're trying to grow that stuff will grow in like 7 to 12 hours, easy, like it grows fast. But if you're trying to culture up a mycobacterium sample, that like you get a sputum test and you can do your acid fast test on that and get your result right away. That's great. What happens if it's inconclusive, but you still think it's probably tuberculosis? You just couldn't get a positive result from your acid fast stain right away. You might want to culture it and give it a chance to grow up and see if there are enough bacteria for that test. Mycobacterium takes like at least a week to grow, and that's on the healthy ones that grow fast. The fast ones will grow in a week. The other ones will take like sometimes months to grow. So, well, that's a problem. So that's prohibitive if you're going to rely on culture. So now you guys probably could think at this point, like, well, then if you think that it is tuberculosis, could you just do PCR? Absolutely. But that's what we're saying here is that phenotypic methods can sometimes be prohibitive if you're relying on culture. They are useful if you're looking for unknowns and you don't know what primers to use for your PCR methods that only take like 3 hours. They're useful if you kind of know what you're already approaching in your biochemical testing and everything like that. So you can look at that sooner and faster than you might need for looking at PCR, especially if you don't have PCR available for that organism, but whatever. So it can be a problem if you have to rely on that. Anyways, this is a little chart that talks about all the stuff we talked about with phenotypic diagnostic for your study purposes later. Your immunological study methods now, testing methods for diagnostics. Immunological is always going to be dealing with antibody antigen interaction. Always. Well, I say antibody antigen or antibody or T cell. Right. We know that. But any sort of antigen interaction and confirming that specific interaction with that antigen, a lot of times it's going to be more closely associated with antibody. If we're looking at serology, this is looking at serum. And so remember, serum is like you get clotted blood And the liquid that comes from that is your serum. In there, we have our antibodies against whatever illness you may or may not have had or actually have. And you might have antigen in there as well. So that's what that's talking about with serum and serology. So we're testing, diagnosing illness based on your serum and whatever antigen or antibody might be in there. It's talking about that specificity between that interaction in your immune system. So you are always going to be looking at this as using a known antibody to look to see if your patient has the antigen, or using a known antigen to see if your patient has an antibody against it. If that doesn't start to sound familiar to you, it should, because that's exactly how ELISA, the direct and the indirect, work. Does the patient have the antigen or does the patient have the antibody? All immunological and serological, that's the same interchangeable terms there for our purposes. All immunological tests are going to be asking that question. Does the patient have the antibody or does the patient have the antigen? All immunological tests are going to be categorized as indirect or direct, always. So that's what this is getting at. Now we can do serology and immunological tests, not just on serum, but we can use antibodies and antigens found in urine, CSF, tissue, and saliva as well. Well, we know that a lot of our serological testing bases off of this common concept, which is simply that if your patient sample, if you want to know if your patient has the antibody, which is the thing on the left here, does the patient make antibodies to HIV? Then you're going to mix it with the antigen and look for a reaction. On the other side of it, what is the identity of the microbe? What antigen is here? So then you can take your patient sample or your culture from your patient sample and mix it with a known antibody and see if there's a reaction. In this case, it's just looking at agglutination and precipitation, which is our most basic reactions that we can look at here. It's pretty commonly used for blood typing. So we're just using anti-A or anti-B or anti-RH antibodies to look for agglutination, essentially, for the blood typing stuff. So this is our summary chart of the stuff we're about to go through. Again, good study tool. So looking at our antigen and antibody interactions, when we learned about ELISA, we looked at our color change that was triggered by the enzyme that's linked on our secondary antibody, right? So if that secondary antibody stuck on, that meant that the thing was there, the antibody or the antigen, and that caused the color change because the enzyme cleaved the substrate. Great. So that's how we visualize that, but that can't always be used in all of our immunological testing. So one way we can look at this is agglutination or precipitation. Agglutination is looking at whole cells, entire cells, and using antibody to stick those cells together. And you'll get pellets associated with those things getting stuck all together. And that can be whole red blood cells, that could be viruses, whatever it is, but we're talking about whole structures. We're not breaking them apart. Whereas precipitation, that's just referring to doing the exact same thing, but with molecules. So we've broken the things apart, essentially. So looking at the antigens by themselves, not necessarily, so spike proteins, not necessarily whole cells or whole viruses. So agglutination reactions, you can have agglutinated mats that will develop, or you can have non-agglutinated pellets that you could see in wells where like the cells will just settle because they're not creating a mat. Or if you're looking at like tubes, that will have to deal with actual pellets in the tube. So it's like, depends on what system that you're looking at, whether you're going to get pellets or whether you're going to get diffuse things or mats or whatever. So I don't need you to focus on whether you get pellets or not, please, because it depends on the test that you're doing, if you're going to get a pellet or a dot or whatever. But the idea here is you're looking for agglutination or precipitation. Just know those terms for me, please. Okay. Whether it agglutinates or precipitates, that's the focus here. All right, and that's all going to be dependent on if those antibodies are recognizing the antigens. Another test that we can do, this is not agglutination or precipitation anymore. Okay, we're moving on to a new thing. This is immunochromatography using antibody-antigen interactions. That's the immuno part. The chroma is dealing with color change. Entography, often deals with something flowing through something else. So in immunochromatography, we have laminar flow, typically of a fluid or fluid suspended sample that's going to flow across either beads that have something attached to them, or in our more common use situations, like you see in the picture, a piece of paper, filter paper, where the liquid's going to flow across it. And when it comes into contact with the appropriate line on the paper, there's going to be a color change reaction. So we're talking pregnancy tests, we're talking COVID home tests, things like that. So you have a color change associated with if that patient sample contacted that line and interacted with it. It's all antibody antigen interaction. Even in pregnancy tests, that works the exact same way. We have interaction of chemical with another chemical, but recognizing whether human chorionic gonadotropin is produced, HCG, interacting with an antibody that has been, triggered to make a color change when it crosses that line. So that's all these tests are doing. That is just a word for what's happening. When you put a sample, it crosses the line and causes a color change. That's immunochromatography. Laminar flow is having that equal flow of that fluid across whatever substrate you're using, whether it is filter paper or a whole like chromatography column of crap. It doesn't really matter. If you guys have ever seen like those videos of people that like cut open like a whole container of liquid, once that liquid starts flowing out of it all in like one big flow, and it looks like it's almost like solid, like it's not moving, but water's flowing out of it, but it looks solid. Like when you get your tap going and the water's just like, looks like it's just sitting there, but water's flowing out because the cup is filling. That's laminar flow. So that's what that's talking about. You want to get that nice, steady, solid flow. If it's uneven, it might not give you even results. Antibody titer is another thing dealing with our immunologic side of things. This is just measuring the concentration of antibodies in a sample. So concentration of our antibodies against, HIV or something like that. How much antibody you have in your system can indicate how recently you were infected. It can also look at past exposures to things that you might not be actively infected with anymore, and also indicative of latent infections like HIV. Serotyping, this is essentially typing different kinds of bacteria based on their antibody response. Some bacteria have markers on their surface, like some kinds of E. coli might have a marker on their surface that other kinds of E. coli don't have. So we can use antibodies against that marker and say, are you this kind of E. coli or are you that kind of E. coli? So then we can have our serotypes that we name based on those antibody interactions. So that's what a serotype is, using antibody antigen to tell the difference now between different subsets of bacteria. Like when we talk about the salmonella, all the different kinds of salmonella that you can possibly have. Some of them have certain markers we can identify with antibody. That could be things like the presence of flagella or capsules or certain markers on the cell wall. Next, we have the Western blot, another immunological test that is commonly used for confirmation. So you have a positive ELISA that shows that you have seen HIV, but maybe the interaction is pretty weak. And so we don't want to rely on just that to tell somebody that they have HIV for the first time. You want to really be sure that you're telling them the correct thing. You might order a Western blot. It's often used for confirmation of other tests, especially when they're weaker. So this is using the exact same concept that we learned with our gel electrophoresis with DNA, but now we're applying it to protein. So you will put your protein in your gel, run it out with your electricity. The big ones get stuck, the small ones go through. Exact same concept using proteins. But now we make it weird, okay? So what we'll do is we will run our proteins out on our little gel. This is our gel, the blue thing in the middle where this first arrow is, okay? Next, now we will transfer it to a membrane. And that's what the second arrow is down here. Putting it onto, and I say membrane, it's like a piece of paper that is attracted to protein. And so you'll transfer it electrically onto that. So that whole picture that you made with your little bands, instead of looking at it in the gel, you're just going to transfer all of that to that little paper. Now you can coat that whole paper with, it doesn't matter what, put it in some milk. And that's honestly, I'm not even kidding, what scientists usually do. They put it in milk. Not kidding. Put that little paper in some milk, and now the whole paper is covered with some kind of protein. But we're only interested in if you have HIV on there, right? We don't want to have accidentally, when we put antibodies to test for HIV, we don't want those antibodies sticking everywhere else on the blank parts of that paper. So that's why you have to block the rest of it. So that's an important step in whenever you do Western blots, because you have to block the whole background so you don't have your antibodies sticking everywhere else where there wasn't protein. Anyways, so you have your gel, you put it on the little membrane, you make sure the membrane doesn't have any blank spots on it by putting it in the milk. Now you can put your anti-HIV antibody, mix it onto your membrane, and it'll only stick to the band where there was HIV protein. Then you can either cause a color change from your colorimetric BS like we do with ELISA, or you can have there's all sorts of stuff you can use, but things like radiation, whatever, take a picture of the radioactive bands or whatever it is to get you a picture of that band. Kind of like with the gels, we had all sorts of different ways you could look at the bands for that too. DNA doesn't just show up. It doesn't have a color. So you have to expose DNA to something to see it. Same thing with the protein. I mean, you're not just going to see bands of protein on a piece of paper. So we just find a way to visualize it. That's the important part there. So run it out on a gel to separate it out, block the whole background, then expose it to anti-HIV antibody, and then have some sort of visualization of it. This is extremely specific because that antibody will only bind to it if there is HIV there. And it can be extremely sensitive as well if there's any band there at all, because we're all consolidating it into that one band. If there's any band there, you should be able to visualize it with your method, whatever you're visualizing it with. So this will be our confirmatory method by consolidating it all into that one band location. Additionally, remember that antibody is only going to bind to it if there's HIV there. It's not like when we look at our DNA bands and we have all those different bands everywhere, because it's looking at any DNA. It's not looking at specific DNA. This is looking at specifically only HIV. You better believe that when we ran that gel, there's just a shitload of other proteins all around in this whole area. But we're only going to be able to see this because that antibody only binds with that. That's what Western blot's for. Great. Yes, it's almost 11, but I'm still going to do it. We're still doing it, guys. Standing up. We're doing the questions, okay? Real quick. Stand up, guys. It's exciting time. Don't you know? All right. I have you guys about lucky because I actually brought the names. The last guys had to deal with, I say lucky, believe me, this is the lucky side of it. They got dice. And so I forgot to bring their little guys until we rolled a dice, and I just called up whole groups, and I had whole groups like have to choose. Yeah, it wasn't pretty. Okay, so Logan, I picked yours first. Which question would you like from the list? Number 1, all right, what type of media is used to encourage the growth of specific pathogens? What's the term for that? Yep, exactly right. So that is a point for Foxtrot. So 1 for Foxtrot. Okay, next. I have Becky.
I'll do 4.
4, easy one. I like that one's pretty straightforward. What is serology?
Does it have an antibody or an antigen?
Exactly. All right, so we got 1 point for that. Alrighty. Next we have Krista is online. I don't know if, because I think I have my speaker on. Maybe I do.
I'm here.
I don't know if you can hear me or if you can interact, Krista. I'm probably not going to ask you because I know you might be busy. I know she's online, but I don't want to have to ask her. So we'll pass on to the next person. This is an empty one. This is another empty one.
I think she answered. Yeah, she did.
Okay, okay. Okay. Cool. Okay, let me pull that up. I can pull it up over here. Let's see. Chat. Okay. The color-based media. Okay. So yeah, Krista, if you wanted to pick one of these questions, we have the biochemical test for number 2 and that. So if we say number 2, number 3, or number 4, which one you like best of that one? Sorry, go ahead.
Number 2, the biochemical test, they're based off of a color change, and they use the Dichotomous key.
Yes, that's right. So yes, so for that that's fine. That's a perfectly good answer. It shows that you're understanding that. I just want to, that's what these questions are for. You guys know that. But for biochemical tests, we identify our bacteria based on our color changes. We can also answer this in a different way, basically looking at it as like our phenotypic tests. Both of those are acceptable answers, right? As long as you understand that looking at those color-based changes are going to be the phenotypic ones. Okay. What, I've put your thing back in the thing. What group were you in, Krista? Bravo? Okay.
Bravo.
Okay. Thanks, guys. All right, there's 2 questions left. I have Melina. Which question would you like, 3 or 5? All right, give an example of a commonly used immunochromatography test. That's easy. Yep. So a pregnancy test, another one that would be an obvious one would be the COVID test. And our last question is number 3, phage typing. I want general answers here. Okay, guys, I don't expect you guys to explain the whole process of it. And I have Anaya. Huh? Yes, I don't mind. Yeah, you can ask. Yeah. Any guesses on the phage typing?
In the ADAR play, and you put the phages on the different sections to identify the different types.
That'll work. Yes. So you guys get the point for that. That you guys were Charlie. Okay, cool. Make sure you guys get your points. Thanks, guys. You can sit down. That's it. Thanks for being good sports. All righty. So yeah, remember the phage typing for those of you that might not have heard Kaylee saying about it. That is going to be where you have your agar plate of your growth. You put the bacteriophages, those viruses, and then see which areas you might be affected by that virus. If it did get affected, that means that that virus only affects salmonella, for example. So that would be salmonella. All right, so next we have immunofluorescence. Again, already first thing kind of highlighted up here I have is direct testing. So we're coming right back to that concept of direct and indirect. Does the patient have the antigen? Does the patient have the antibody? All of these tests rely on that. So immunofluorescence uses antibody that it's not linked to an enzyme like ELISA is. Instead, it's linked to literally a fluorescent molecule, and that can be any kind of color either. And so computers specifically can tell the difference by analyzing it, but you can also just use any fluorescent marker and look at it under a microscope. So when we look at this picture that is depicted in our example here, these are actual bacterial cells that got coated by a fluorescent antibody that was antibody anti to syphilis. So we're looking here at syphilis cells, those little spiral shaped cells that are coated in anti-syphilis antibody that are fluorescent antibodies. So that's all that's happening here. This is telling you that that person has the antigen because our antibody against syphilis recognizes that syphilis was there in that sample. It's a lot simpler than you might think it is. Don't overthink it. So we can look at that in a microscope. We can also look at things in microscopes using that other side of the concept, that indirect. Does the patient have the antibody? But here you might be using patient. So you take, let's say you think the patient has HIV. So you put HIV antigen on a slide, in a tube, it doesn't matter, whatever it is, HIV somewhere, antigen, put the patient sample on that. If they have the antibody to HIV, it'll stick. Then you put another antibody that sees human antibodies, and that fluoresces. So it's just using another layer. It's using a secondary antibody like we do with our indirect testing in ELISA, but again, fluorescent, not enzyme linked. Make sense? All right, so direct or indirect fluorescence. The next one is ELISA. I don't think I really need to go over it. I hope I don't have to go over it. This is using that enzyme that'll cause that color change, asking the question for the indirect, do they have the antibody? And then of course we have our little picture to depict that. You put the antigen in, you put the patient sera, does it have the antibody or not? And it follows down positive and negative on each side, the negative being on the right. But you put the sera, If it has the antibody, it'll stick to the antigen. Then we'll put our secondary antibody. That'll get stuck to that. We add our substrate, and then there's a color change or not. You guys know how it works. The next one is the direct ELISA. Do they have the antigen? That's our sandwich method. It gets less pictures for some reason, but it's here we're adding our antibody, putting the patient sample in that may have actual COVID virus in it, for example, and then another antibody that recognizes COVID that's attached to an enzyme, substrate, color change. It's exactly what we learned in lab. Then you'll look at our samples in our 96 well plate, and we don't usually have just 2 rows. It's usually going to be pretty full because we're usually going to be testing for different variations, different dilutions, and all sorts of stuff, different antigens that are on the surface of these things, and looking at to see if that person is positive or not based on color change. The complement fixation test is another immunologically based test. It's immunological, even though we're looking at here complement, which we know is an innate response, it still depends on specific antibody antigen interaction. But we're going to see if you have any antibody antigen interaction based on whether or not complement lyses or doesn't lyse red blood cells. So essentially, we're going to have our patient serum added in with known antigen. If it binds, complement will recognize it. And then our sensitized red cells will be added. There won't be any excess complement there though. So we should have binding of complement and antibody and these sensitized red cells, and then they will settle at the bottom of the tube, as opposed to if there's no antibody to interfere with all of that, leading to lysis. Don't worry about which one's positive and negative. I'm not going to give you an example of a positive or negative test and whatever. Just understand that this test relies on the complement interaction with antibody. That's all you need to know. Okay. Classical pathway, right? That's what you need to know about it. It relies on that. Because I know that one gets confusing. So I don't want you to worry about the results of it. Just know that it deals with the classical pathway. In vivo testing, you can do actual immunological tests in living tissue. We know this because when you go get your PPD skin test for the tuberculosis test, we are looking at living tissue on your body to see if you have an immune reaction. This is T cell related, right? This is not antibody related, but it's still an immune reaction. So we talked about that in the last one. You guys love specificity and sensitivity. It's your favorite thing of all time. It's not confusing at all. So specificity versus sensitivity. Again, I always start with sensitivity because if you're really sensitive, you're going to be affected by any little amount of something. So the idea of sensitivity is trying to pick up as many positives as possible. Even the very weakest ones, you'll see that they're there because you're so sensitive to them. So you are making sure you get up all of the positives, so you do not call something negative when it really is positive. You want to get all of the positives. That's sensitivity. Specificity is just a different thing. It's not against it. They work together. Specificity is looking at it as I'm only going to see the positives. I'm not going to accidentally pick up negatives and call them positives. So with specificity, you're only seeing positives and not accidentally calling the negatives positive. So low false positives, because remember, a false positive is calling something positive if it is not positive. If you're specific, you wouldn't do that. That's against your religion. So if you're very specific, you're not going to want to see anything as positive when it's not. If you're sensitive, you're going to make sure you get all of the positives so you won't call something negative accidentally. So low false negative on those. All right, now onto our genotype stuff. These are pretty straightforward because they're all just looking for the presence of the DNA sequences or how somebody might react. Again, when we look at the forensic stuff, cleaving with the restriction endonucleases, that can be used in diagnostics as well, but it's not as commonly used as PCR and things like that, okay? So if we're amplifying nucleic acid, there's something called nucleic acid amplification test. I guarantee you almost all of you, when you put your talks together for your personal presentations, which are going to be due, soon. Yes, those, when you guys are going through your diagnostic methods, this is why I highly recommend that if you guys can at least get a rough draft into me on time. I'm sure you guys know you're going to be presenting on your chapter day. So if you have fine tuning to make onto your slides, be sure that that fine tuning is done by your actual presentation day. I'm not grading shit until you present. Okay. So if you want to submit on the due date, because I know that's before your presentation days, I have to have everybody submit something by the due date. So everybody's on the same time frame. But if you submit something that's your rough draft that gets across like what you are wanting to present, okay, as long as that has all your information on it, even if it's not got your pretty pictures and whatever, that's fine. And I'll look over it and let you know if it's acceptable, because the number one mistakes that people make on these presentations is whenever I'm asking for your diagnostic method, people will just say they use urine samples and blood samples. Absolutely inappropriate for this class. If I'm sitting here talking to you about PCR and Western blot and all of this stuff, you're expected to tell me if they use PCR or Western blot or immunochromatography or whatever to test for your specimen. Do not submit a slide to me that says urine sample. Okay, you will get points off for that. That's the number one point loss location. Number two point loss location, treatment methods. If they really only treat because like if you have a disease that there is no treatment for, or you have a cold, come on now. Like what are they gonna give you for a cold? They're gonna tell you to push fluids and go take some Tylenol. Like you can put that, that's okay. Like if that really is, but if you're telling me about something like COVID, where 99% of us might go home and push fluids and take Tylenol, but if you're in a hospital for it, they're going to give you these medications or this antiviral or whatever, and all you're doing is like, go home and drink fluids. That's a problem. So treatment methods are another thing. Be sure you're giving specifics on that, because if we're talking about antibiotics and learning about exact antibiotics, I expect you to tell me exact drugs that are used. Some people don't have any problem with that. They'll copy and paste it, and that is fine, as long as you guys understand how it applies to your illness. Not that I'm going to necessarily ask you questions. I'm usually more lenient about the question asking on the presentations, as long as you guys are, when you're up here telling me about your illness, only using note cards and slides as references. If you're reading straight off of your slides or you're straight off of your note cards, you will lose points. If you are putting a slide up that has all of these tests on there, many of which we never talked about in class, you better be ready for me to ask you a question about those tests. If you're putting some up there that we don't know about, you're good. Like you're fine. As long as you put some up there that we learned about and you can explain them. If you have a caveat on there of my disease has weird tests or something, that's understandable too. But if you're just like copy pasting everything and it becomes very clear to me that you just copy paste stuff on there, that's where your problems are going to come into play. You understand what I'm saying there? It's just like with your teams, if you guys put up slides up there that nobody can explain anything on there and they're just reading it, that's where the problems come in. You're supposed to know it well enough that you can teach it. Know it well enough that you can teach it, right? But also keep it brief. Okay, don't make it too long. We're trying 5 minutes or less, please. Because you guys are going to present your presentations before I talk on the lecture. So let's say it is sexual transmitted disease week or day, and we have someone coming up talking about herpes. You guys will give your herpes presentation, and then I will give my talk about the sexually transmitted disease, and I will not be talking about herpes, and you will still be expected to know everything about herpes based on what that person presented on. So when I say that you have to explain it as well as I would explain it, I literally mean that, because I will not be lecturing on that topic if you are talking on it. So be aware of that. Okay. Yes.
I thought when I'm looking at the schedule, it just says that we're just doing the presentation. I'm not showing that we're learning a new chapter on presentation.
For unit 3, we aren't. That's group thing. Your individual presentations are unit 4. So for unit 4, every chapter that we have for skin diseases, if you chose a skin disease, you're presenting on skin disease day. Congratulations, you'll be the first ones going. So that's all unit 4. So it doesn't say that on there, that's presentation days. You guys are going to get an email next week with literally everybody's names on what day you'll be presenting. That's based entirely on the disease that you chose. That is absolutely why I do not tell you that beforehand, because I don't want people all choosing sexually transmitted diseases. That's ridiculous, because that's the last day, right? So I don't tell you that on purpose. And that's why everybody's is due before we go into unit 4, so that you have at least your information out there. And I can tell you, hopefully beforehand, that you need to add more information on your treatment stuff. Yes. So that's why we have it that way, to benefit all of us. But yes, everybody will be presenting. I had a student come to me yesterday asking, do I actually have to present this in person? Yes, there's no caveat, there's no getting around it. So especially at this point in the semester, maybe if somebody had asked earlier in the semester, I might have changed things, but nobody ever does. They always wait till it's due. So no, you have to present in person. Their other option that I do sometimes give people is for you to record it. And some people will think that, oh, okay, well, in recording it, then I get out of having to do this in front of people. Then you get to sit there while I play your recording in front of the entire class, and you get to watch your recorded face and voice in front of everybody, which usually is worse. Because if you just have to be up in front of somebody, you don't have to see yourself doing it. I mean, you might hear it coming out of your mouth, but like seeing yourself up, I promise you it's worse. When I go back and look at all my recorded lectures, you guys, I hate it. There's a reason why I don't look at them. So, so yeah, so in person, just come up here. I've told people in the past, sort of like with your group stuff, if you put a lot of information on your slides, that covers your bases, guys. Have it on your slides, and you don't necessarily even have to bring up every little detail on your slides, but it's there covered. So you can just like summarize your slides. Come up here and be like, well, when they usually treat COVID, most of the time we're getting fluids and Tylenol and told to push that. Sometimes they will treat it with antiviral ribavirin or something. And then, but you have all the other information up there talking about the details that the doctor might give and other viruses, antivirals that are sometimes used, but you don't have to talk about every little thing on your slides. Summarize. Summarizing is fine. So when you guys do that, just keep that in mind. Especially if you're up here talking like I do usually about my slides, and I'm just like, here's the slide and here's what I'm saying, and I'm not reading off of it, I'm using it as a prompt. That shows that you guys understood what you put on your slide. That's what I want. Show me that you know what you made. Anyways, what I'm trying to say is nucleic acid amplification test is going to show up on almost everybody's slides. It will. NAATs. If you've been putting your slides together, you've probably seen NAATs and been like, sure, that's a test. And what it means is nucleic acid amplification tests. And that's almost always going to be PCR based. Not all of them will be, but most of them are. Then we have hybridization, which is like our probes, making sure that DNA sequences match other DNA sequences. And when they do, they bind together, and you can use even fluorescence tagged to that probe to see it. And then we have entire genome sequencing. We know PCR, we're just making copies of the DNA. You have to know what DNA you're looking for in order to give it the right primers. There's different kinds of PCR, real time, where I tell you that the A, the T, the C, and the G are associated with different fluorescent colors. And when they bind on, it releases the color. The machine reads it the whole time. And you can literally see how the machine is reading each color and how it's progressing. Because if you're making copies, you're going to be releasing colors. If you're not, you're not. So then you can look in real time while it's going and running what is happening. Reverse transcriptase PCR is just taking a RNA sample and turning it into DNA and then doing regular PCR with that. That's all that is. Multiplex is looking at various primers associated with more than one disease. So this is how we can do one swab from you and put it into a PCR machine and look for flu and COVID and RSV in one PCR test, which is something that they can do nowadays. It's pretty new technology, but that's all in one test. They have to be specifically designed that way, and you can't do it with a lot of things. Pan-bacterial is basically a qualitative PCR just to see if you have gram-negative or if you have like enteric bacteria genes, but it's not going to give you an idea of exactly which organism. But if you want to have some place to start, it's a good test to do. I don't know what RT lamp is, so don't ask me. Hybridization, again, the probes themselves that recognize sequence for a specific, toxin or some aspect of that pathogen. It's made out in that sequence, attached to maybe a fluorescent thing. We take the cells, break them open, mix it together. If it binds, you see the fluorescence. If it doesn't, then there's no fluorescence. Just like with the antibodies, right? We have that specific antibody antigen antibody linked to the fluorescence. It makes it glow. Instead of antibody antigen, we're literally looking at the ATCG binding instead. So this is literal DNA specificity. One of these examples is FISH, which is fluorescent in situ hybridization. So this uses that concept using whole cells. So we can use like tissue samples, biopsies, and things like that without having to have liquefied or broken open cells and look at the actual tissue in like a microscope to see if there was fluorescent binding going on. So it's easy to confirm diagnosis like that. This is an example of FISH where we have a patient blood sample that was a blood culture where they grew, I don't know, some sample from the person's blood, put it on a slide and gram stained it. They say, oh, it's gram positive, it's cocci, it's staphylococci, but is it Staph aureus? So what they'll do is on that slide, put that fluorescently labeled DNA sequence that recognizes Staph aureus only and mix it on that slide and then look at it to see if it sticks. And in the microscope, you can see that fluorescence. And that's how that works, just like the antibodies. It's the same thing. It's just we're looking at DNA to DNA instead of antibody to antigen. Okay. That's FISH. Whole genome sequencing is so fast nowadays with like the primers that we have developed for just looking at random sequences that if you have an outbreak going on that you need to diagnose quickly, doing an entire genome sequence is easier than trying to figure out what it is up front, maybe, because you have so many people that are involved. You can even see drug resistance involved in certain organisms with that as well. Microarrays use that same concept of the FISH thing, but so instead of, so you think that your patient, let's say we're looking at cancer, okay? Your patient has breast cancer, but you don't know why their breast cancer is breast cancer, like what genetics are causing that. So you can take a microarray chip that has on all these little dots, 365 little dots on this chip, different DNA probes. And those probes will each bind to different mutations associated with different forms of cancer. Take your patient's sample, DNA that you've isolated from that patient's biopsy, let's say, mix it with that chip, and then put it in a machine, and it tells you which spots that DNA bound to. So you can look at different mutations that are associated with different cancers. You can look at different subtypes of bacterial species if you're looking for infection. You can have chips on there with DNA for all sorts of different bacteria and say, yes, it bound with this one, it's probably this, and then do more tests to confirm. So microarrays can look at a large, wide variety of genetic information all on one chip. And it's hybridizing, just like FISH, and usually uses fluorescent labels based on whether it bound or not. And then a machine analyzes it. You don't have to. We can use this concept. You can see very easily how if you're using this concept mixed in with maybe your ideas about how the antibody antigen thing works and create chips that are designed to look at, okay, this person has GI illness. Let's put them on lab on a chip for GI illness. It'll look for genetic issues with the GI illness, as well as antibody antigen interactions with pathogens, all sorts of stuff on one chip. Put that patient's fecal sample on there, pop it in a machine, and it'll give you results saying it's more likely that this is Crohn's disease, or it is more likely that this is salmonella. This is really particularly useful for like labs, I say labs, but medical situations in rural areas, as well as like underdeveloped countries where they don't have labs available. And we can provide these things to them so that they can at least do testing on patients and get them the treatment that they need. So that's the idea with the lab on chip. And it can also now starting to use CRISPR technologies dealing with altering genetic sequences for certain purposes for the chip's sake. Yes, other ones, these are in our other category now. We're starting to get into that gray area. Mass spec, and we're almost done. Mass spectrometry. This is an example of a mass spectrometry technique that is MALDI-TOF. I don't remember what MALDI stands for, but I remember what, I know M is mass, okay, but TOF is time of flight. And what happens with this is they shoot these lasers at the patient sample, whatever it is. And what happens is ions will fly off and it'll keep shooting at it. And the machine measures the ions that fly off, how long they spend in that vacuum, time of flight, their time of flight in the vacuum, as well as our mass, literal like molecular mass. and another aspects about charges, things like that, about these ions, right? And as they're made, the machine is able to analyze all of that information and then tell you the literal exact molecular makeup of whatever it is in that sample. Your patient comes in and they've tried to commit suicide by ingesting a drug, and nobody in the family has any idea what it is. They just know that there was some kind of drug involved. And so you take that person to the ER, you can take their blood sample and run mass spec on it, and it will give you the exact chemical composition of what is in their blood. So you can determine what drug it was that they've tried to overdose on. So that can help with treatment plans for that. It also can be used for identifying microbes, because certain microbes have certain things maybe on their surfaces or whatever it is that helps you identify that as well. So MALDI-TOF is for the molecular level of these things and identifying them that way. It can even give you a sense of how heavily infected somebody is based on how many ions you get. All right, so next we have the obvious ones like imaging, MRI, CT, X-ray, PET scans, finding areas of localized infection or tumors, whatever it is that you're trying to diagnose. So you can do imaging that is less invasive. And now you know where to do biopsies, to do aspirations of maybe areas where you have granulomas or you have tumors or areas of infection. So if they have an abscess somewhere, aspirating samples there as opposed to having to do exploratory surgery. So obvious ones there, imaging.